Hello, everyone, and thank you for joining us. In this presentation, I will address some practical aspects and pitfalls in the molecular diagnosis of brain tumors. The increasing understanding of the molecular biology of brain tumors led to the incorporation of molecular parameters into their classification for the first time in 2016, which was a paradigm shift from morphology-based into integrated diagnosis of many brain tumor types. At the same time, technological advances decreased costs and increased availability of molecular testing, which has been quite dramatic for sequencing technologies after the introduction of next-generation sequencing, or NGS. For these reasons, molecular testing became part of the routine diagnostic workup of many brain tumor types, such as adult diffuse gliomas. Three types of molecular parameters have been incorporated into the 2016 classification so far: mutations, gene fusions, and copy number variants, or CNVs. And they can be evaluated using four main testing platforms. PCR-based and sequencing assays are the platforms of choice for detection of mutations and gene fusions. Although they can also detect CNVs, FISH and array are the primary platforms for the evaluation of this molecular parameter. FISH and array can also infer the presence of gene fusions, but are unable to define the specific gene fusion events. In general, mutations and CNVs are tested using DNA, whereas gene fusions are tested using DNA or RNA. All molecular testing platforms have factors that impact testing in the pre-analytical, analytical, and post-analytical phases. These are some factors that I will discuss today and show clinical cases primarily in the context of NGS testing to illustrate pitfalls in brain tumor molecular diagnosis that we have encountered in our clinical practice here at Mayo Clinic. Prior to testing the pre-analytical phase, there are two critical factors. The first one is tumor purity, which is the fraction of tumor cells present, and it should be above a minimal level to avoid a false negative result. As an example, for somatic heterozygous mutations, which are mutations present in one copy or allele of a gene only in the tumor, NGS testing of a sample with low tumor purity will generate less mutant sequencing reads than a sample with high tumor purity. If the analytical sensitivity of the test, which is the test's lower limit of detection, is of 10% mutant sequencing reads or variant allele frequency, a mutation in a sample with 10% tumor would have 5% variant allele frequency, leading to a false negative result. Tumor purity is decreased by the presence of non-neoplastic cells, including normal brain and inflammatory cells. Hemorrhage typically does not impact DNA-based testing as mutation analysis, since red blood cells are enucleate and lack DNA. The second pre-analytical factor is tumor amount, which is the total number of viable tumor cells present, and it should be above a minimal level to generate a valid result. For example, in NGS testing, even in samples with high tumor purity, low tumor amount may generate insufficient total number of sequencing reads, also known as depth of coverage, which is typically required to be of at least 100 reads or 100x coverage, leading to a failed result. This is particularly problematic for large NGS panels, as well as whole exome and whole genome sequencing, which evaluate hundreds or thousands of targets simultaneously. Tumor amount is decreased by the presence of tumor necrosis and in small biopsy specimens, which substantially lower amounts of nucleic acid are extracted when compared with resection specimens with similar tumor purity. The first case to illustrate pitfalls in brain tumor molecular diagnosis was the left frontal tumor resection of a 50-year-old man that was histologically consistent with a glioblastoma, showing areas of abscess-like suppurative necrosis with abundant neutrophils. As this patient was younger than 55 years and the IDH1 immunostain was reportedly negative, IDH testing is recommended and was performed using our neuro-oncology NGS panel. This panel interrogates 219 brain tumor-related genes, including IDH1 and IDH2, and consists of a DNA panel to detect mutations as low as 10% variant allele frequency, and an RNA panel to detect gene fusions and transcript variants. In our laboratory, we perform tumor enrichment by manual microdissection whenever possible. In this case, the areas of abscess-like necrosis with abundant neutrophils were accidentally included for macrodissection, reducing tumor purity to only about 20%. The NGS panel showed the absence of an IDH mutation and an NF1 mutation at a variant allele frequency close to the analytical sensitivity of the test, without mutations typically observed in IDH wild-type glioblastoma, such as third promoter mutation. These findings prompted repeat testing to exclude the necrotic areas and increase the tumor purity, which increased the frequency of the NF1 mutation, uncovered a PTEN and third promoter mutation, and confirmed the presence of an IDH mutation, showing that this tumor indeed had the classical mutational profile to support the integrated diagnosis of an IDH wild-type glioblastoma. This case illustrates how inflammatory cells may substantially decrease tumor purity and lead to false negative results. Case 2 was the biopsy of a left temporal ring contrast enhancing tumor on a 66-year-old man that histologically showed an atypical infiltrating astrocytic proliferation without mitotic activity, microvascular proliferation, or necrosis, consistent with a diffuse astrocytoma that was likely not representative of the highest grade of the lesion. The IDH1 immunostain was negative, and third promoter as well as IDH NGS testing were performed to determine if this tumor could be potentially upgraded to a molecular glioblastoma based on the CIMPACT Now Update 3 recommendations. In this case, both the tumor amount and tumor purity were limited. By NGS, a third promoter mutation was observed at 4% variant allele frequency, which was slightly below the validated analytical sensitivity of the test and was called negative. By droplet digital PCR, which is more sensitive than the NGS with an analytical sensitivity of less than 1%, the low-level third promoter mutation was confirmed. No IDH1 or IDH2 mutations, even at low level, were identified by NGS, with all sequencing reads mapping to the reference consistent with an IDH wild-type genotype. In this clinical pathological context, the absence of an IDH mutation and the presence of a third promoter mutation supported the integrated diagnosis of a diffuse astrocytic glioma, IDH wild-type with molecular features of glioblastoma, illustrating the importance of using high analytical sensitivity platforms when testing cases with low tumor purity and low tumor amount. Other strategies to optimize testing of cases with low tumor purity include selection of a tissue block with the highest tumor fraction when multiple blocks are available, as well as tumor enrichment by macrodissection as we perform in our laboratory. For cases with low tumor amount, testing can be optimized by increasing the number of tissue sections and combining multiple tissue blocks for testing, as well as prioritizing testing when multiple tests are indicated. In both scenarios, it is important to utilize testing platforms with high analytical sensitivity, which is indeed one of the main analytical testing platform-related factors that impact testing. Among the four testing platforms, PCR-based assays and FISH are the ones with the highest analytical sensitivity and usually evaluate only one to a few targets with low tissue requirements, costs, and turnaround time. Sequencing and array technologies, on the other hand, usually evaluate multiple targets more comprehensively at the expense of lower analytical sensitivity and higher tissue requirements, costs, and turnaround time. Case 3 was the right parietal tumor resection of a 73-year-old woman that was histologically consistent with a glioblastoma showing pseudopalisading necrosis, and the IDH1 immunostain was negative. Although the integrated diagnosis of an IDH wild-type glioblastoma could be rendered without additional genetic testing, the neurology NGS panel and 1p19q codeletion FISH were performed. While the NGS panel showed the absence of an IDH mutation, as well as a third promoter, EGFR, and PTEN mutation, further supporting the diagnosis of an IDH wild-type glioblastoma, FISH showed loss of one signal of 1p and 19q, consistent with 1p19q codeletion. The molecular findings in this case appear to be discrepant, as the 1p19q codeletion is typically not observed in the context of a classic glioblastoma morphology and an IDH wild-type genotype. This apparent discrepancy, however, can be explained by a limitation of 1p19q codeletion testing by FISH. The FISH testing probes cover only a small portion of 1p and 19q arms, and therefore are unable to distinguish partial arm from the whole arm codeletion that is diagnostic of oligodendroglioma. In addition, combined losses of portions of 1p and 19q arms spanning the FISH testing probe locations, which would be called positive by FISH, may be observed in diffuse astrocytic gliomas, including approximately 3% of IDH wild-type glioblastoma. To determine if this was the case, chromosomal microarray was performed and showed decreased signals involving 1p and 19q terminal regions that span the FISH testing probe locations, consistent with partial arm rather than whole arm 1p19q codeletion. In addition, copy number abnormalities typically observed in IDH wild-type glioblastoma, such as EGFR amplification, CDKN2A and B homozygous deletion, and chromosome 10 loss, including the PTEN gene, were observed. So, taken together, the overall molecular profile of this case indeed supported the integrated diagnosis of an IDH wild-type glioblastoma, illustrating the importance of being aware of testing platform limitations to avoid potential misdiagnosis. After testing in the post-analytical phase, there are a few factors worth discussing. In tumor testing, two types of genetic alterations are detected: somatic and germline variants, as illustrated here in the context of NGS testing. Germline and somatic sequence variants are variants that result from changes in the sequence or spelling of a gene. Somatic variants are genetic alterations that occur only in the tumor cells, not in normal cells, including germ cells, and therefore are not inheritable. These variants include the mutations that we are intentionally targeting in tumor testing. As somatic variants are present only in the tumor, the variant allele frequency of these variants is proportional to tumor purity. Germline variants, on the other hand, are genetic alterations that occur in tumor cells and normal cells, including germ cells, and therefore are inheritable. For the most part, germline variants are not clinically relevant and considered incidental findings in tumor testing, with some exceptions that I will discuss later in this presentation. As germline variants are present in all cells, the variant allele frequency does not vary with tumor purity and is usually at 50% when present in one copy of the gene in the heterozygous state, or 100% when present in both copies of the gene in the homozygous state, unless there are associated gene copy number abnormalities. Tumor testing may be performed with paired normal tissue, allowing for distinction between somatic and germline variants, as the variants identified in the normal tissue are germline variants that can be filtered out from all variants identified in the tumor to determine the somatic tumor-specific variants. Most clinical laboratories, including ours, however, perform tumor-only testing and are unable to definitively distinguish somatic from germline variants. To assist in the evaluation of variants that are sequence variants identified in tumor testing, a multidisciplinary group compiled guidelines to standardize interpretation and reporting in 2017. Based on the therapeutic, diagnostic, and prognostic implications, the strength of available clinical and experimental evidence was stratified in 4 levels. Using these levels of evidence, sequence variants identified in tumor testing can be classified in 4 tiers. Tiers 1 and 2 variants are variants that show some level of evidence supporting a pathogenic role in cancer and are mutations that should be reported. Most of these mutations are somatic. However, some may be germline, as approximately 10% of cancer genes harbor somatic and germline mutations. When germline in origin, these mutations are often associated with hereditary cancer predisposition syndromes, and these are some genes associated with brain tumors that fall into this group. Tier 4 variants, on the other hand, are variants that do not show cancer association and have been reported in 1% or more of normal individuals. These variants are classified as benign or likely benign and do not need to be reported, as they are thought to represent normal sequence variation, also known as single nucleotide polymorphism. Lastly, Tier 3 variants are variants that do not qualify as either pathogenic or benign and are classified as variants of uncertain significance, or VUS. Case 4 was the resection of a left frontal glioblastoma on a 52-year-old woman that was negative by IDH1 immunostain. Based on the patient's age, IDH testing is recommended and was performed using our neuro-oncology NGS panel, which showed an EGFR and a third promoter mutation, in addition to an IDH1 variant involving codon 236. The structural characteristics of this variant did not support any significant functional impact, and this variant has been observed in normal individuals, but at less than 1% frequency. Based on somatic databases and literature, there was no evidence supporting cancer association. So, taken together with currently available data, this variant was classified as a variant of uncertain significance. As this IDH1 VUS does not carry the same clinical implications of an IDH1 R132 mutation and no IDH2 mutation was identified, the IDH genotype for this tumor was wild type, which, in conjunction with the presence of a third promoter and EGFR mutation in this clinical pathological context, supported the integrated diagnosis of an IDH wild-type glioblastoma. This case illustrates how to interpret and integrate the finding of a variant of uncertain significance in brain tumor molecular diagnosis. The last case was a right cerebellar tumor on an 8-week-old girl that histologically consistent of a mitotically active proliferation of rhabdoid cells. Immunohistochemically, the tumor cells showed loss of expression of INI1, consistent with complete biallelic inactivation of the SMARCB1 gene, as well as a polyphenotypic profile with EMA and smooth methyl-actin expression, supporting the integrated diagnosis of an atypical teratoid rhabdoid tumor. Given the patient's young age, the neuro-oncology requested molecular profiling using our neuro-oncology NGS panel as well as chromosomal microarray. In this case, both the tumor amount and tumor purity were high, and the NGS panel showed a truncating loss of function SMARCB1 mutation that has been recurrently reported in atypical teratoid rhabdoid tumor involving all sequencing reads at 100% variant allele frequency. Chromosomal microarray showed a relatively stable genome with loss of one copy of chromosome 22, wherein the SMARCB1 gene is located, as the only abnormality. The monosomy 22 was assumed to be somatic and present only in the tumor, as germline monosomy 22 is extremely rare and associated with phenotypic changes that this patient did not have. Therefore, the SMARCB1 biallelic inactivation in this tumor occurred through loss of one copy as part of the monosomy 22 and mutation of the remaining copy, which could be somatic and present only in the tumor, not in the normal, or germline and present in the tumor and in the normal. If somatic in origin, the SMARCB1 mutation variant allele frequency in the context of high tumor purity would be high, but less than the 100% because of the presence of wild-type sequencing reads coming from both normal gene copies. If germline in origin, however, the SMARCB1 variant allele frequency would be close to 100%, as the mutation would be present in all sequencing reads except for the reads coming from the single normal gene copy, suggesting that in this case, the SMARCB1 mutation was likely germline in origin. Whenever a medically relevant germline mutation is identified in tumor-only testing, follow-up testing on a normal sample along with genetic counseling should be recommended to determine the mutation origin. In this case, the mutation was confirmed to be germline and was consistent with the diagnosis of a rhabdoid tumor predisposition syndrome for this patient, which can occur in approximately 35 to 40% of cases of atypical teratoid rhabdoid tumor. This case illustrates that occasionally mutations identified in tumor testing may be germline, especially in pediatric patients, and that tumor-only testing may suggest a germline versus somatic origin based on the mutation, variant allele frequency, and the gene copy number status. Another important post-analytical factor is the genetic nomenclature, which for human genes and gene sequence variants follows the recommendations of the Human Genome Organization. Occasionally, commonly used and recommended genetic nomenclature are discrepant and may appear confusing in molecular test reports. For example, the name of the KIA 1598 gene, which is a partner of a recurrent FGFR2 fusion reported in the recently described polymorphous low-grade neuroepithelial tumor of the young, has been updated to SHOOTING1, as the KIA designation is a placeholder symbol to be updated when the protein function is identified. Another example is the nomenclature of the third promoter mutations, which was coined by initial research studies and consists of a combination of the three last digits of the genomic coordinates of the recommended nomenclature at the genomic DNA level, flanked by the nucleotide changes of the recommended nomenclature at the coding cDNA level. In conclusion, genetic testing plays a central role in brain tumor molecular diagnosis. There are multiple testing-related factors that impact genetic testing, which are important to be aware of to avoid molecular diagnostics pitfalls as illustrated in this presentation. I would like to acknowledge my colleagues who form our molecular neuro-oncology team here at Mayo Clinic, and on behalf of our team, to thank you for your attention. Please do not hesitate to contact me should you have any questions.